Okay, so what we're looking at is a sheep brain dissection specimen. This brain has the meninges intact. So what you see here that looks kind of iridescent is the dura mater. You can also see where the dural partitions are beginning to form, and this would be where the confluence of the sinuses is. Cerebellum is here. Spinal cord is exiting here. If we flip it over to the inferior view, you can see where the olfactory bulbs are, and this is one of the olfactory bulbs left in place. Our optic nerves from the left and right sides are here. You can see where it's forming the optic chiasm, that little X-shaped region just anterior to the pituitary gland. So this large bump here represents the pituitary. Here you can see the pons and medulla oblongata before the spinal cord forms. So what we want to do is we want to make a longitudinal cut through the longitudinal fissure so that we can separate both cerebral hemispheres from each other. So make a gentle cut and basically open this sheep brain up like a book. Now keep in mind, a sheep brain and a human brain are not color-coded like a model is. So it may not be perfect, and everything may not be as easy to visualize as you might think they should be. Alright, so now we've got our hemispheres open. You can see everything is a shade of brownish gray. So from this point, you can see how clear the dura mater is and how tough and thick and protective it is. So I'm going to take the scalpel, open the dura mater. Now the arachnoid mater, because it is like a spider web, usually as you pull back on it, you may be able to see some remnants of it there, but it will snap and we'll lose it as we open this up. Now we can see the surface of the brain. You can see how it looks glossy. This represents the pia mater. The blood vessels that you see here in brownish, kind of black color, are deep to the pia mater. And you can tease it up and peel it off to see how matte the tissue looks in color deep to it. But it just comes off very, very small pieces. Now on the sheep brain, like the human brain, we have specific gyri, or ridges of tissue, and fissures in between to create that wrinkled look. We're not going to worry about discerning lobes and specific gyri on the sheep brain. So we've cut as close as we can get to the longitudinal fissure, and as we do that, we're going to compare it to the human brain model. So I'll prop it up back here. A little bit better. Yep. There we go. So on the sheep brain, what we see here is our corpus callosum, which corresponds to this. You can see it is a definite lighter shade because it is white matter full of axonal tracts with our gray matter organized on the outside. And you can see it a little bit clearer. We'll make a second cut in a moment to further demonstrate that. Inferior to the corpus callosum, we have our fornix, another arching band of tissue. And between those two are septum pellucidum. So you can see the septum pellucidum here looks like a little membrane. But if we punch behind it and take it away, we're in the lateral ventricle behind there. If we go back to our fornix and look inferior to it, and I'm going to maneuver our brain down a little bit, you can see the rounded structure of the thalamus. So this is our thalamus, and the hypothalamus would be here. Unfortunately, in most sheep brains, you cannot see the funnel-like structure very clearly. May be able to tease it back a little bit here to take off the excess tissue. Most of the time it comes out relatively unclear. Tease off a little bit. So here's our thalamus, our hypothalamus, which leads down to the pituitary gland. Now in these sheep brains, which have been sitting flat, they're kind of pushed to the back, and here's our infundibulum that holds the pituitary gland onto the hypothalamus surface. From the thalamus, if we look posterior to it, we should see a little tissue tag, and we do see that here. This is the pineal gland, which is responsible for our sleep-wake cycles. And from there, as we work our way down, we start to see the brain stem. So it's broken into 3 different parts. The proximal part, and I just cut it a little bit, didn't mean to do that, is the midbrain. Within the midbrain, you have your cerebral aqueduct, which is this little line here. So we said our lateral ventricle was here. Our third ventricle sits in the midline between the thalami, and the cerebral aqueduct connects the third ventricle to the fourth ventricle that lies here. Also, as a part of the midbrain, if you look on the posterior aspect of it, you can see that there are 2 distinctive bumps. There's 2 more on the other hemisphere. These are called the corpora quadrigemina, and these are responsible for auditory and visual reflexes. The middle part of the brain stem, the anterior facing bump, is known as the pons, and inferior to that, until we get to the foramen magnum, it continues down as the medulla oblongata. Finally, on this view of the sheep brain, you can see that the cerebellum is organized in a similar fashion as the cerebrum, with white matter on the inside and gray matter on the out. The white matter organization in the cerebellum looks kind of like a tree, so it's called the arbor vitae, which means the tree of life in Latin. All right, so now what we want to do is just take a look at that gray and white matter organization by making a coronal incision. So I'm going to continue my coronal cut through this brain specimen and open it up to show that there is a distinctive organization of gray on the outside and white matter on the inside. Again, white matter is where your axons lie. The white color is due to the fatty deposits of myelin around the neuronal axons. And the gray matter are where the dendrites and the cell bodies are going to be found.